Please confirm me. Am I, am I audible and the screen is visible, right? Quickly confirm. Am I audible and the screen is visible, right? Just type yes or no. Okay, perfect. Okay, every one of you quickly answer my question. My question is, what is the name of the bond that is formed between the sugar and the phosphate group? See, we are discussing a very small basics only. We have to read extremely depth, then only you can learn it. What is the name of the bond? Very good. No, Aishwarya, we should not call passport diester. I told you, right? It's called anhydride or the ester bond. Always keep the point in your mind. Phosphodiester bond is the bond which is formed between the two nucleotides, between the OH group as well as the 3' hydroxyl group and 5' phosphate group. That is called as the phosphodiester bond. Very good. So, friends, in the yesterday class, we just read some basics. For 1, 2, 3 classes are little basic. Now we have to go little advanced. Okay, then only you can learn the depth of the concept. Okay, now listen to me very carefully, everyone. What are the basic rules of the DNA replication? In the yesterday class, before ending the class, I told to all of you very clearly that it is mandatory that the DNA has to follow every single rule of the DNA replication. If it miss any kind of a rule, the DNA never undergo the process of replication. Now, the first one, we called it as what, friends? A semi-conservative model of the DNA replication. What is this semi-conservative model? Why the DNA has to follow this model? What is the significance of this particular model? Listen, in the yesterday class, I have very clearly explained it to all of you. Watson and Crick, when they proposed the double helical structure of the DNA, they got an idea. What idea they got? They got an idea that they have to propose a model. By using that particular model, the DNA is undergoing the process of the replication or the duplication. So what model they proposed? Semi-conservative. In the yesterday class, I told to all of you friends, what is a semi-conservative model exactly? Listen, every one of you. According to the semi-conservative model, both the parent strands are getting separated. You can see that. When both parent strands are getting separated, there will be a synthesis of what strand? There will be a synthesis of daughter strand, which means that one parent strand and one daughter strand. This is the semi-conservative model. And what I have told you, most of the scientists accepted this model. Some of the scientists opposed this model. They did not accept this. By opposing this model, they proposed some other models. We called conservative model. The third one is called, friends, a dispersive model. What did I told you about the conservative model, friends? If you quickly recall, according to the conservative model, what did I told you? The entire parent DNA is getting preserved and it is giving rise to the daughter DNA. And what you remember, what is the dispersive model? Dispersive model is called as a cut and patch model. What is this cut and patch model, friends? Small daughter, small parent, small daughter, parent, daughter, parent, daughter, parent, daughter, parent. This is called dispersive model. Now you tell me, what is our ultimate goal right now? Our ultimate goal right now is we have to prove what we have to prove. We have to prove that semi-conservative model is the idealized model for the DNA replication. You have to prove this. At the same time, we have to disapprove. What we have to disapprove? We have to disapprove conservative and dispersive model is not followed by the DNA. It only follows the semi-conservative model. To prove this, the two intelligent scientists named as Matthew Messelson and Franklin Stahl, they have done a beautiful experiment. As I told you in the yesterday class, this is one of the most classical experiment that have ever done so far in the field of biology. Now let me explain you. I want every student has to remember this all the time. If you remember this, understanding this particular experiment becomes easy. As I told you, out of 12 question papers, 6 to 8 times, the question were asked, especially in the part C from this particular concept. I mean to say the experiment. So that's the reason it is very important. You have to know the concept here. Nothing to read, nothing to remember. Everything to be understand. If you understand the concept, you can answer the given question easily. Now listen. What is semi-conservative model? Don't forget this. According to the semi-conservative model, the parent strands are separated and acting as the template strand. Is it? And the point is that, friends, it's also very clear. There is a synthesis of daughter strand. Now every student listen. Which is called as heavy strand, which is called light strand. Every student all the time remember, friends, parent strand is always called as heavy strand. And daughter strand is called as light strand. So parent is heavy and daughter is light. But what you have to understand, in this particular diagram, you can observe parent strand also, daughter strand also. Then what do you call it this as? You can call it as intermediate. Okay, what do you call it? Intermediate. Sometimes instead of using the word intermediate, they will use the word hybrid. So anything is same. In the exam, if they give intermediate also, if they give hybrid also same. So we have to prove now. What we have to prove? We have to prove the DNA contains one heavy strand and light strand, and it always follow the semi-conservative model only. I will explain the experiment slowly. I will show you the wonderful animation. You will get a very, very good idea. Listen. In order to prove this experiment, first what the Messelson and Stahl has been done, listen, they have taken a test tube. Okay. So friends, in this particular test tube, they have kept a bacterial culture, E. coli bacterial culture. And this entire culture, they kept under N15. What is N15? You know, right? Heavy nitrogen. So from this particular E. coli, friends, listen, every one of you listen. They have isolated the DNA. What? They have isolated the DNA. When they isolated the DNA, remember everyone, they started observing the base. When they start observing the base, they found what? It's a heavy DNA. But what is our goal according to the semi-conservative model? We should get intermediate. We should get the combination of heavy and light. But what do we got in the first time? We got only light. Sorry, we got only heavy. Next time what they have done? Once again, they have taken the test tube. They kept the E. coli culture. Remember everyone. And this time they kept the E. coli culture under the N14. Once again, what they have done, friends, listen to me carefully. Once again, they have isolated the DNA. And what they started doing now? They started observing the base. When they start observing the base, this time what they got? They got light. See, friends, first time they got heavy. Second time they got light. But what we require intermediate. Now, next time what they have done, friends, next time they have taken a test tube. To this particular test tube, what they have done, they have added equal concentration of N15. They have added equal concentration of N14. Along with the N15 and N14, they have also added cesium chloride, which is a salt. Now you have to ask me a question, sir, why we have added only cesium chloride? There are many salts are present. The reason is that the density of the DNA and the density of the cesium chloride, which is almost equal, which is 1.91 gram per centimeter cube. That's why we have added only cesium chloride. Now, friends, what we have done, then we did the process of centrifugation. What is centrifugation? You all people know that rotating at a higher speed. You know how much time they have rotated, friends? They have rotated up to 20 hours, not 1 or 2 hours. They have rotated up to 20 hours. So now you tell me, when they rotated up to 20 hours, how they're going to settle? Of course, friends, they will always follow the density, which means they will always follow the weight variation. So who is going to settle down? Undoubtedly, cesium chloride will settle down first because it is having higher weight. Then followed by N15 will settle. Correct, na? Then at last, which is going to settle? N14 is going to settle. Friends, with this simple idea, this is the idea. What is the idea? Yes, we can separate the heavy DNA, we can separate the light DNA based on the density gradient or based on the weight variation. They got this beautiful idea. Yes, we can separate heavy and light based on the density. With this simple idea, they started observing, they started experimenting all the models. They started checking all the models actually. Now, friends, listen. Now, you all people understand this is a simple one. Now, they started doing all. Now, first to first, what they have done, same thing. Listen, everyone. Listen, everyone. First, they have taken the test tube. In the test tube, what they kept, friends? They kept only N15. They have isolated the DNA. You only tell me when they isolated the DNA, what they got. You look into this. They got original DNA, which we also called as a heavy DNA. But what is our goal? We should not get heavy, right? We should not only get heavy. We should get heavy and light, which we can call intermediate or hybrid. This is what we have to get. So we did not get that in the first time. First time, what do we got? We got heavy. Next, what we have done, a little amount of the N15, all the students has to understand, a little amount of the N15 has been transferred to the N14. The moment they transferred to the N14, from there, every 20 minutes, they are started taking the sample. Now, the question is very clear. Why? Why only every 20 minutes? Why not 30 minutes? Why not 10 minutes? Why not 40 minutes? Because we are doing the experiment with E. coli. Every student commonly know that the generation time of E. coli is a standard time, which is the 20 minutes. That's why, friends, we started taking the sample every 20 minutes. Now, all the students try to understand. At the first 20 minutes, you only look into this. We can call it as, this is called generation 0. This is called generation 1, generation 2, generation 3, and so on. In the generation 1, you tell me what they got. Did they got heavy? No. Did they got light? No. What they got? Neither heavy nor light. But what they got intermediate. That is what we require, right? That is the semi-conservative model. What is the intermediate? Combination of light, combination of heavy. That is what exactly we needed. In the generation 2, what do we got? We got 50% light. We got 50% intermediate. Friends, it doesn't matter whether the light DNA is repeating or not. What is important for us? Intermediate DNA is repeating or not. In the third generation, 75% is light. And what do we got? 25%. 25% is intermediate. In the fourth generation, 12.5%. In the next generation, 6.25%. So what's the point? The point is that every single generation, which type of DNA is getting repeated. That's the important point. Which is getting repeated? Undoubtedly, semi-conservative is getting repeated. Here we got intermediate, which is the semi-conservative. Here we got the intermediate, semi-conservative. Here we got intermediate, which is the semi-conservative. Intermediate means combination of heavy and light. Hence, friends, listen my words. 2 generations are sufficient. 2 generations are sufficient to prove. What to prove? Semi-conservative model only the idealized model for the DNA replication process. You only think it. If it is a dispersive model, what we discussed here, entire parent DNA is getting preserved and converting into daughter DNA. Does it happen anywhere here? No, not at all. What is the last one? Dispersive model, cut and paste. Small daughter, small parent, small daughter, small parent. Have you observed anywhere in any generation, small daughter, small parent in any generation? Have you observed? No, you have not observed. So what's the point? The point is that every generation semi-conservative only getting repeated. Hence, they proved that semi-conservative model only the idealized model, the DNA is always going to follow the semi-conservative model only. Now, I am just pasting one link in the WhatsApp group. I want every one of you go back, watch that animation and come back immediately. It's a 6 minute of animation. It's a really, really good animation. I want you to watch it right now, immediately, quick, and get back to me once it's done. Everyone, start it. I have already given the link. Everyone. Once it's done, just type it. Just let me know once it's done. Just let me know once it's done. Let me know once it's done. Okay, is it clear, everyone? If you have any kind of a doubt, I will re-explain to all of you. I hope you understand the experiment very clearly. Quick, 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 quick. Others, quickly. Okay, perfect. I hope the experiment is clear for every one of you. Okay. Not to hurry, friends. Just, just be calm and answer the question. Look into the question. In the Messelson and Stahl experiment, what part of the DNA gets labeled with 15N? I want every one of you to answer the question. What part of the... Why only thymine, Sakshi? Anyone else? You don't need to tell the thymine. We generally say that base. Okay. So it's not necessary that the thymine only has to be, identified. We generally say that base. Base is the part that is getting, highlighted or the getting labeled with the nitrogen. Is it? Yes. Okay. I want all the students has to think and answer the question. I want everyone to respond. Okay. This is not a theory class or it's not a meditation class. If you compete with others and if you compete yourself, then only you will be successful in the exam. Okay. It's all about the super competition. Don't sit quietly. See the question. In the Messelson and Stahl experiment, where on the cesium chloride gradient would the following DNA found? They're asking, friends, where all these are all found. Which is found in the middle, which is found in the low, which is found in the high. Tell me, Ranjita. Just answer like this. Suppose 1 is in the low, 2 is in the high. Type all together in one option. Meghala, everyone. Archana, Kavya, what's the answer? No, don't type like that. Just type all together in one option. Suppose 1 is at low, 2 is at high. Everyone. Mini, what's the answer? 1 is... No. Nancy, that's what I'm telling you. Slowly you have to see the question. We did not ask the question which are heavy or which are light, dear. We are asking the question their position, which they are situated, where they are got located. That's the question. You have to tell me the places. Sharmila, what's the answer? Naveen? See, friends, the question says that where all these are all found. DNA double-stranded, where both DNA strands are labeled. You tell me, friends, when both strands are labeled, it contains weight, right? So which is called heavy. Then heavy, where it will found, friends? Heavy will always go to lower side. Now, look into the second one. DNA double-stranded, where only one strand is labeled. One strand means what? It is having a intermediate weight. And where it is located? It is located in the middle. Third one, double-stranded DNA, where neither strand is labeled. No strand is labeled. It is very light. Which is very light, it will be present at which position? High position or the first position. Everyone got it clearly or not? Everyone understand this correctly or not? They are just asking the positions where they got located. Every one of you, very clear. Right. Good, good, good. Not to, what is the name of this Samsung SM? Please keep your names. By that I can, I can remember easily. Okay, that's great. That's great. That's great. Okay. See, since we just started 1 or 2 days, that's all. The moment when I finish 3 to 4 units of your syllabus, 3 to 4 units, then you will understand what is the real CSIR exam is, how depth you have to study, what kind of way you have to study. So my only humble request to all of you, because from the last 7.5 years, I am seeing many students who are super excited for the 10 to 15 days. Okay. These kind of students never qualify the exam. Once again, I'm telling you how excited you are in the first day of your preparation. The same excitement you have to show it in the last day of your preparation. This is the real quality of qualifying the competitive exam. See, 1 or 2 days you will get a gap. That's a common point. That's no need to worry about that. But you have to show your consistency in order to qualify the exam. I am telling you, your hard work only required, intelligence is very little. Intelligence is required only while analyzing the questions. Anyway, listen. So we understand right now the first rule of the DNA replication. What's the first rule? The first rule of the DNA replication is said to be the semi-conservative model. And friends, what will be the second rule of the DNA replication? As we already studied that, friends, the DNA has to follow every single rule compulsory. If it miss even one rule also, it can't undergo the DNA replication. What's the second rule? It should starts at origin. Friends, what do you mean by that? It should starts at origin. All the students remember, the process of DNA replication should not start randomly. The process of DNA replication should start always at a one particular place, at one particular position. The point or the place at which the DNA replication will start, we people generally call it as the ORC, origin of replication. But in case of eukaryotes, we don't use the word ORC. Instead of using the word ORC, in case of a eukaryotic organisms, friends, we will use the word called as replicator, not to get confused with that point. Now, we all know very well, if you look into the E. coli, which is the prokaryotic organism, what will be the shape of the DNA? We all know that the DNA is the circular in shape. Correct, na? So all of you try to understand me. So the circular DNA, and you know that, friends, prokaryotic DNA is a very small DNA. So how many number of ORCs are present, friends? There will be a presence of only single ORC. Origin of replication is only one in case of prokaryotes. And friends, most of the time for the prokaryotic organism, for the prokaryotes, which organism we will mostly take as a better example, friends? You all people know that we take E. coli as the very better example. Is it? Yes. Similarly, friends, when you are reading the eukaryotes, it's also a very good point. Whenever you are reading the eukaryotes, why do you take yeast as the best example? Why can't we take any other as the best example? Because, friends, we are having humans having 23 pairs of chromosome. Yeast is having 16 chromosome. Some are less or equal, not very equal. Some are less or equal to the humans. That's why we will take the yeast as the best example when we are reading the eukaryotes. In case of a yeast, all the students listen my words without confusion. In case of prokaryote single origin, we can call ORC. In case of eukaryotes, we generally call it as replicator. Why? Because multiple ORC. This is one ORC, this is another ORC. That's why we can call it replicator. In case of where, friends? In case of eukaryotes. The same replicator having the other name, where in case of a yeast. Whomever joined newly, please continue. The same replicator in the yeast having other name, we called it as ARS. What is the full form of ARS? Autonomously replicating sequence. Not to get confused with that particular point. I want you to remember that point very correctly without having any kind of a confusion. So that is the second rule, starts as the origin. The most important, the significant rule, very important rule. We have to understand this rule very clearly, friends. Here only you have to focus. You have to understand very nicely. In what direction the DNA replication will occur? From the plus intermediate level or from the +2 level, we always use it to read this statement. The new strand of the DNA has to be synthesized only in the 5' to 3' direction. There are many reasons are there. I will explain you. One of the simple reason, but this is not the major reason. One of the simple reason I am talking, you know, right, which enzyme is the major enzyme involved in the DNA replication? We all people know that DNA polymerase is the major enzyme involved in the DNA replication. You people also know very well DNA polymerase enzyme is going to read the parent strand. Friends, I am talking about parent strand, okay? It is going to read the parent strand in 3' to 5' direction. Friends, if it is reading the parent strand in 3' to 5' direction, undoubtedly, friends, it is going to read the daughter strand in which direction, friends? It is going to read the daughter strand in 5' to 3' direction, not to get confused with that particular point. But this is not the major reason. What will be the major reason? All the students try to understand me very correctly without having any confusion. Just focus. We know very well in the yesterday class we studied how the phosphodiester bond is getting formed. Suppose, friends, this is nucleotide, this is the nucleotide. What we studied, friends, in the yesterday class very clearly, the the nucleotide at the sugar. The sugar at the third carbon, what group it contain? It contain OH group. You all people commonly know that. The OH group, where what did I told you? Oxygen acting as a nucleophile. What I told you? Oxygen nucleophilically come and cut one phosphate group. Then two phosphate groups are releasing out. Then there is a formation of the phosphodiester bond. That every one of you know it very well. Now, we have to prove. What we have to prove? Why the DNA replication has to happen only in the 5' to 3' direction? Why not? Why the DNA replication should not happen in 3' to 5'? This is very interesting, friends. All the students listen. Every one of you listen this. This is the 5' to 3'. Here what group is present? Tell me. At the 3', you all people know that every time OH group is present. Yes or no? Of course, yes. Follow me. This is what, friends? A nucleotide. In the yesterday class, I have told to all of you very clearly, friends, whenever the nucleotide is coming, the nucleotide is always comes. Every student has to understand my points very correctly without confusion. The nucleotide always comes with how many phosphates? The nucleotide always comes with 3 phosphates. Never ever get confused with that. So now this OH group will cut one phosphate, releasing of 2 phosphates. Now the nucleotide is added. No problem. You got it very correctly. Now, friends, you know, right? We will discuss in the next class. The DNA polymerase enzyme start doing the proofreading. Proofreading means what, friends? Checking the errors. It will start check the errors in which direction? It is going to check the errors in 5' to 3' direction. So, friends, during the proofreading also called as exonuclease activity. Anything is same. During the proofreading or exonuclease activity, the DNA polymerase enzyme is identified. What it identified? Whatever the nucleotide that it is added, it is a wrong nucleotide. It is not a healthy nucleotide. Then what happened as a part of the proofreading mechanism? It immediately removes that particular wrong nucleotide. Of course, that's fine. Now, the moment it removed the wrong nucleotide, friends, it has to replace with the healthy nucleotide. Yes or no? Of course, yes. Undoubtedly, it has to replace with the healthy nucleotide. How it is going to replace? See, once again, this is the nucleotide. Again, here, what group is present, friends? OH group is common, right? Nothing happened to the OH group here. Now, once again, this nucleotide will come. OH group will cut one phosphate, 2 phosphates will be released out. Then, friends, nucleotide is added. No problem. So, high energy bond is cleaved, providing the energy for polymerization. Such an important statement it is. What the statement is saying that? The statement is saying that there is a cleavage of the bond. Whenever there is a cleavage of the phosphate bond, then only, friends, there will be a formation of the phosphodiester bond. That is possible only when there is a presence of OH group. So now we understand very clearly the synthesis of the new strand has to happen only in the 5' to 3'. Friends, now we have to disapprove, friends. What we have to disapprove now? We have to disapprove why the DNA replication should not happen in 3' to 5'. You only look into the picture. Now 3' to 5'. Every student know, right? At the 5' direction, how many phosphate groups are there? 3 phosphate groups are there. Everyone know it very clearly. Now, friends, you only listen. Listen, just understand. Don't think that it will happen in the body. It is an hypothetical model. We are proving the DNA replication should not happen in 3' to 5'. So now you look into this. This is the nucleotide. All of you try to understand me. Here the nucleotide contain what group, friends? OH group. Yes or no? Yes. Now this oxygen will go and cut the one of the phosphate group. How many phosphates are releasing out? 2 phosphates are releasing out. Yes. Now this entire nucleotide will attach here. There is no problem at all. Everything is fine so far. Now, during the time of the proofreading, what has happened, there will be a removal of this particular nucleotide. Okay, now we removed this nucleotide, it came out. Now, friends, you tell me. Now, I want to add this nucleotide here. Okay, here the OH group is present. Now, every student tell me, do you think this OH group can go and cut the phosphates here? Is that possible? No. Why? Because there is a no presence of the 3 phosphate groups here. Now you tell me, do you think this OH group can go and cut the phosphodiester bond? No, because there is a no presence of the phosphate group only. Because whatever the phosphate groups are present, in the first time only we got cut it. Then next time we remove the nucleotide, then there is a no presence of the phosphate group. Hence, friends, you can't add this nucleotide. Why? Reaction does not proceed because high energy bond is not getting cleaved, because there is a no high energy bond only. That's the reason, friends. The DNA replication never ever happens in 3' to 5'. It always happens in the 5' to 3'. Not to get confused. If anyone got confused, listen. First, at the 5' direction, there are 3 phosphate groups are present, which are very common, right? And all the students listen to me very correctly. This is the nucleotide having the OH group. This one, with the help of oxygen, will go and cut the phosphate. Then 2 phosphates are released out. Now this entire nucleotide with how many phosphates? 3 phosphates will come and added here. But during the proofreading, we understood that this nucleotide is a wrong nucleotide. Then we removed it out. When we removed it out, now there is a no 3 phosphates, right? No 3 phosphates. Now we are replacing with a healthy nucleotide. But how can we replace? If we want to add this nucleotide here, first we have to cut 1 phosphate here. There is a no phosphate group only. Then how can we cut? That's why nucleotide is not added. But here there is a presence of the OH group here. Then it can easily cut the phosphate group of the new nucleotide and it can add the nucleotide very easily. Hence, we can prove that the new strand or the daughter strand has to be synthesized every single time in 5' to 3' direction only. Never ever get confused with that point. Now, very, very unique rule, very interesting rule, uni or bidirectional. What do you mean by the word unidirectional? Friends, listen my statements very correctly. Whether it is a prokaryotic organism or whether it is a eukaryotic organism, friends, the DNA replication never ever starts randomly. As I already told you, whether it's a prokaryotes, it has to start from origin. Whether it is a eukaryote, which is a linear DNA, it has to start from the origin only. All the students, please look into this diagram. Suppose example, in case of a, let's talk about the prokaryotic organism. Let's consider. Suppose this is the origin. This is the origin. The replication fork is moving only in one direction. You can see that highlighted area. So this is called as what? Unidirectional replication. What is a bidirectional replication? Now you can see here, this is the origin. So one strand is moving in one direction, sorry, one fork is moving in one direction, another fork is moving on another direction. This is what do we call, friends? This is we call it as a bidirectional replication. So friends, what's the interesting point? The interesting point that every student has to understand is whether it is a prokaryotic organism or whether it is a eukaryotic organism. Every organism, mostly bidirectional replication only will happen. Most common, not the unidirectional replication. Very important statement it is. Asked statement in the exam. So now you have to ask me, sir, what do you mean by the word replication fork? I can tell you in a simple words, replication fork is the point or is the place at which active replication starts. The point where the active replication is starting, that point only we can call the replication fork. Every student, please look into the point. Suppose the two parents, the two parent DNAs are separated and each one is giving rise to what? Daughter strand. Is it or not? We are going to read in the upcoming slides. Just listen to me. One is always move towards the replication fork. You all people know very well. One is moving always away from the replication fork. This also every student commonly know about it. That I will explain in the next slide itself. Now, where in case of eukaryotes I am talking about, whenever the two strands are opened, you can see, right, it has given a Y-shaped structure. This Y-shaped structure only we can call the replication fork, not to get confused with that. And for the exam point of view, you remember that whether it is a prokaryotic organism or eukaryotic organism, bidirectional replication is the most common replication. And the most important point among all, the most important step is this one, semi-discontinuous replication. See friends, I am explaining you rule only, not the process. While reading the process, we will explain utmost depth. Now we are only reading about the rule, semi-discontinuous replication. So all the students, please look into the diagram very carefully without confusion. What is our rule? We are having a rule that the new strand has to be synthesized every single time. In which direction, friends? Every time it has to be synthesized in the 5' to 3' direction only. That's the rule. Now you look into the picture. Now there are two parent strands. One parent strand is in which direction? You only tell me. One parent strand is in 3' to 5' direction. There is no problem in that. Everything is smooth now. No worries about it. What about the other strand? So here daughter strand is synthesizing in which direction? 5' to 3' towards the replication fork. According to our rule. So it passed our rule. But what will be the next strand? Next strand, it is located in the 5' to 3'. But every student know very well, DNA polymerase enzyme is reading the parent strand in which direction we studied. It is going to read in 3' to 5' in this way, opposite. And what is other rule? Other rule is that daughter strand has to be synthesized in 5' to 3' compulsory. So that's why this is moving in this direction and other daughter strand moving opposite. You can see that. Which means, listen to me, in the process automatically you'll get a good idea. One strand is always moved towards the replication fork. Now you have to ask me, sir, what do you mean by the word towards the replication fork? Whichever the direction helicase is moving, in that direction it has to move. So this is called towards the replication fork. And this one, see, another strand also undoubtedly synthesizing in the 5' to 3' according to our rule. But what's the point? It is moving away from the replication fork. This is towards, this is away. So which is moving towards the replication fork, which is moving continuously, we can call it leading strand. And we call that particular synthesis as a continuous synthesis. Whichever is moving opposite, what do we call it as? A lagging strand, right? And we call it this particular synthesis as discontinuous synthesis. So friends, you have to understand me. Why do we call discontinuous? What is the reason? Because it is moving away from the replication fork, that is fine. But while it is synthesizing, it is always synthesized in the form of a pieces. Small pieces. In the process, you will get a good idea. So since it is synthesizing in the form of a pieces, that's why we called them as the lagging strand. And these pieces given the name called as Okazaki fragments based on the name of the discoverer. Based on the name of the discoverer, we called it as the Okazaki fragments. Not to get confused with this particular point, I want you to listen this very correctly and do not confuse. Okay, you got that. Now you look into this beautiful picture. Now you'll get a good idea. So this is the one parent strand which is moving in the 3' to 5'. You can see very clearly here that daughter strand is moving in 5' to 3' continuously towards the replication fork. That's why I call leading strand or the continuous synthesis. Now you look into the red color strand. So now you see the daughter strand. Daughter strand, look into this. Daughter strand is synthesizing in 5' to 3', no doubt about it, but not continuously, in the form of a pieces. In the process, I will explain you why these pieces will come. Why not continuously? So this pieces, based on the name of the discoverer, we can call Okazaki. I'm requesting all of you to remember the length. Listen, all of you. One Okazaki fragment, only one Okazaki fragment in prokaryotes. It is very long, 1,000 to 2,000 bases. One Okazaki fragment. But total number will be very less. Hardly you can say that 10,000 will be number. Total number are very less. But length of one Okazaki fragment is very high in prokaryotes. In eukaryotes, length of one Okazaki fragment, friends, 100 to 200 nucleotide. Very short Okazaki fragment in eukaryotes. But you know, friends, how many number it contained? It is present in millions and millions of copies, not one or two copies. So Okazaki fragment in eukaryotes, small in size, more in number. In prokaryotes, large in size, less in number. Simple point. Then the next rule, unique rule, RNA primers are required. You all people has to think it correctly. Okay. See, whenever you are reading molecular biology, cancer biology, biochemistry, then techniques, it is all about our understanding. Never try to, imagine, never try to by heart something. See, suppose when we are reading ecology chapter, when we are reading evolution chapter, there are a lot of things to be remembered. But these concepts are conceptual. You require a wonderful understanding of the concept. Now you listen. Why do we require primer? Let me tell you one good example to all of you. I don't know whether you remember these examples or not. In the childhood, you might observe in the villages. Right now also you might observe in the villages. In the villages, we are having a borewell pump. You know what is the speciality of this borewell pump, friends? If you want to get the water from this particular borewell pump, first you have to pour some water. I don't know whether you have seen it or not. You have to pour some water in the borewell pump. When you pour some water, when you pump continuously, then the water will be coming continuously. Is it? Those are all the borewell pumps which we use to observe. Now the same thing will happen here also. You tell me, what is the function of a DNA polymerase enzyme? Once again, all the students has to understand me. The DNA polymerase function is what? Adding the nucleotides. What do you mean by the word addition of the nucleotide? One nucleotide has to be added with the second one with third one with fourth one. So friends, if the DNA polymerase enzyme wants to add the nucleotides, first of all, someone has to come and add some nucleotides. I hope you're getting my point. Someone has to come and add some nucleotide. When someone adds some nucleotide, then the DNA polymerase enzyme continues that nucleotides. Now you can ask me, you ought to ask me a very good question, sir. Then why not DNA polymerase enzyme only cannot add that nucleotides? Why we require someone else? So because DNA polymerase enzyme do not have a primase activity. Simple question, simple point. It do not have a primase activity. So there is an enzyme called as RNA primase. What the RNA primase will do? It will come and add the primer. Now, if you ask me the definitions are what is primer? See, friends, primer means small group of RNA nucleotides. Simple definition. Small group of RNA nucleotides. What is the length of this primer in our body? In our body, it is 11 plus or minus 1. That's all. But in PCR, in a laboratory, it is up to 60 nucleotides. Anyway, that's not the matter. But the length of primer is what? 11 plus or minus 1. So RNA. So now you have to ask me one more question, sir. Why we are adding RNA primer? Because we are synthesizing DNA. Because we don't have a DNA primer. We don't have a DNA primer. That's why we will add RNA primer. But anyway, at the end of the replication, we will remove it. We know that very well. We will see in the process. We add the RNA primer. The moment we add the RNA primer, the polymerase enzyme will come and continues the nucleotides. In case of a leading strand, in the process, you will get it. In case of a leading strand, friends, you are going to add only one primer, that's all. But friends, what about in the lagging strand? In the lagging strand, we are going to add multiple primers. All the students remember, leading strand one primer, lagging strand multiple primers. In the option, in the process, you automatically get it. Okay. Now, not to get confused, slowly think and answer the questions, even though you know the answer. Even though you know the answer, think it. So we always, what the answer we can give? GC. Why? A and T contain only 2 hydrogen bonds. G and C contain 3 hydrogen bonds. So here low energy is required. Friends, here higher energy is required. Correct, na? So the answer will be the G and C. Good. Everyone has to answer. Don't sit silently, please. Because no one is writing exam on behalf of you. You have to write the exam. Okay. Yes. Question number 2. Slowly think and analyze and give the answer. Okay, Naveen, given so fast. Number 1. Everyone, others, Mini, what's the answer? Sharmila said 3. Okay. Others, Nancy, Sakshi, everyone. What's the answer? 4. Okay. What is the name Samsung? Can you please tell me the name? By that I can remember. Okay. Next. Okay. All the students, please look into the given question. See, I am telling to all of you one important statement. Remember my words. Okay. Always remember this till the end of your CSIR exam. See, friends, reading is different. Okay. Listening the lesson is different. Revising the concept is different. But answering the question is always a skill. Okay. The skill can be enhanced when you are understanding the question correctly, when you are analyzing the question correctly. When you don't analyze the question correctly, you never answer the question very correctly. Okay. It's all about analysis. The exam is all about analysis. Now look into the first, the question says that. What the question says that? What is the best way of answering the question? The best way of answering the question, make it simple. Simplification is the ultimate way of answering the question correctly. The question says that a segment of the lagging strand of the DNA molecule having this sequence. This is the point. So this is one parent strand. This is another parent strand. This is called leading strand and this is called lagging strand. What is the point question given? The sequence of the lagging strand is this one. A small segment of the lagging strand having this sequence. 3 prime, AG, CG, AT, GT, AC, GC, 5 prime. Okay, this is the first point according to the question. What is the second point according to the question? According to this, what should be the sequence of the DNA that is template for the complementary leading strand of this segment? See, friends, they have not asked the question, what is the template for this? Listen. So, if it is having 3 prime to 5 prime sequence, what about this? It is having opposite. So every student is answering this. That is not the question asked. They are asking the question, what is the sequence of the template of the leading strand? This is leading strand, right? So what is the sequence of the template strand? This is they are asking. Then here also what sequence will come? 3' to 5' sequence only will come, right? Which means the same sequence will be repeated. Hence the answer is what? Answer is 3. Every student understand this or not? Everyone give the reply. Is that clear? They have not asked the sequence of the leading strand. They have asked the sequence of the template of the leading strand. Ranjita, you understand clearly? So that's why while we are answering the questions, we should be very slow. We should analyze that very correctly. Analysis is very important. Okay, perfect, perfect, perfect. Not a problem, not a problem. Okay, that's all for now. I'll see you in the evening sharp 7 p.m. through the Zoom as well. Okay, through the Zoom only. Okay, friends, that's all for now. I'll see you in the evening class. I'm requesting you, please continuously revise it. Okay, thank you so much. Thank you all. Thank you. Thank you.